we could address that end of the day. Also, we also looked at elite controllers as well, and then it's really cool. We still pick up the virus signals present in, especially as I mentioned, one of the major signals we're seeing the NALT. Really, that's the first time basically the imaging technology allowed us something associated with mucosal tissues where we see majority of signals, and even we saw something in eyeballs. There is a virus lights up.
Thank you. Another question, please.
Hi, Tina Peachman from MHRP. My question for you is, besides blocking the alpha-4 beta-7, these are activated T cells. Are you signaling by using your antibody? Have you looked at anything downstream? I mean, you're using it to block the alpha-4 beta-7, but are you actually stimulating other immune responses in your T cells?
Right. We looked at some of the regular T cell activation markers and all. We don't see really coming up any higher level than the alpha. There will be no much differences. Also, we looked at a lot of, especially chemokines and cytokines. That's one of the issues. We don't see that any major changes actually. That's pretty well works with okay.
Thanks.
Thank you.
Did you look to the frequency of other immune cell subsets after the alpha-4 beta-7 treatment in the tissues that you analyzed?
Yeah.
Alpha-4 beta-7 can be expressed by other cells.
What tissue, what subsets you thinking?
I think alpha-4 beta-7 can be expressed by NKs and maybe
Right.
macrophages. That
Yeah,
may,
we will look at all these the battery of tests, battery of markers actually. There is a major differences, but there are not much difference, especially some of them. But all we know is there is not major drawbacks with this antibody. Most of the cells get preserved.
Thank you.
Thank you very much, Dr. Barai. Please join us here
Okay.
at this stage in case we have at the end some general questions.
The next speaker will be Michael George from University of California, Davis. The title of his talk is Oral Microbiome in HIV-infected Women, Aging, Disease Progression, and Opportunistic Infection Increase the Pathogenic Profile.
Hi. I'd like to start off by thanking the conference organizers for inviting me to share a little bit of my data with you today. I'm going to start off with this graph that shows what I think everybody who's paying attention to the microbiome has already noticed, that we've had this exponential growth over the last 10 years in the number of microbiome studies, and it actually coincides, not surprisingly, with the development of this pyrosequencing technology. But I want to point out that it's appropriate, I think, that the number of publications and studies of the microbiome and HIV infection kind of mimics this pattern. And just in the last couple, 2 or 3 years, as Jason was referring to, we've seen several studies that point to a role for the microbiome in chronic inflammation and immune activation, as well as microbial translocation. And let's not forget that the appearance of opportunistic infections themselves represent massive disruptions or dysbiosis in the microbiome. And even more recently, there's been some speculation that the microbiome may play a role in the development of HIV-associated non-AIDS conditions, which are very much akin to the process of aging. Unfortunately, though, if you look at this number of oral microbiome presentations or studies over the same period, you see a striking lack of exponential growth in these studies. And this is quite surprising, given what we already know about the oral microbiome from non-HIV studies. The bacterial community structure is very much, I don't want to say similar, but like the same. It's represented by the same types of phyla and genre that the gut microbiome is. There's some advantages to the oral microbiome that include, like the gut microbiome, it's a fundamental part of human development and health and disease. Unlike the gut microbiome, however, it's an easily accessible sample. It's repeatable. You can take many samples during a day longitudinally without any problem. It's a highly structured yet remarkably resilient community, and it's also an excellent model for both bacteria and host bacteria interactions. Like the gut microbiome, there are some disadvantages. Namely, there's a large number of uncultivated, uncharacterized species, and there's an enormous amount of interpersonal variability in the taxonomic distribution. This actually underscores the very high likelihood that there's going to be multiple pathways to dysbiosis from person to person. So in the backdrop of this paucity of information on the oral microbiome and HIV infection, I decided to develop a collaboration with Doctors Barbara Weissler and Harold Berger and the Women's Interagency HIV Study to start taking a look at some of the saliva samples that they've been biobanking over the last couple of decades, actually. So we developed a concept sheet, which is a pilot study application, in which we threw in an extra little twist, and we were interested in the impact of aging in all of this. The CDC now estimates that due to the baby boomers and the success of art, that over 50% of the HIV-infected population in this country is now over 50 years of age. So we hypothesized that there will be an increase in pathogenic bacteria and fungi and viruses that will be more elevated in the aging population than in young adults. And so in AIM-1, we're just going to do phylogenetic comparisons of uninfected and infected young and aging populations. And in AIM-2, we're going to do some PCR analysis of viruses and fungi that are involved in the opportunistic infections of the oral cavity. So we got 36 samples, and this is a breakdown of the groups we devised from those samples. We actually had to drop out a couple samples because we had a range of ages, and then we decided to define the age groups more clearly, naming the young group as being under 35 and the aging group as being over 50, which dropped out a couple samples. But you can see we have some decent representation in a number of groups, albeit very low for statistical analysis. We also designed the study so that the CD4 depletion was similar in each group, as well as the viral loads. This is the workflow for our phylogenetic analysis. We extracted DNA, surprisingly, with the MOBio Power Soil Kit, and we did paired end sequencing of the V3, V4 amplified region, and we did that on an Illumina MiSeq system. So I'm going to cut right to the chase because the time is short. These are the averages of the groups, and what we found was dysbiosis in both related to aging and HIV infection. And I'm just highlighting some of the things that I'm going to talk about in the next few slides. At the phylo level, there was an increase in Bacteroidetes and Proteobacteria. And when we looked at the genus level, the Bacteroidetes was actually mostly represented by Prevotella. And there was indeed more changes than we could see at the phylo level in the Firmicutes, and these were represented by Veillonella. And in the Proteobacteria, most of the changes we saw were Neisseria. Looking at the patients on ART and the patients with opportunistic infections, we saw a lot of the similar types of changes in phylogenetic profiles, in that increased Prevotella was predominant, and also changes in Veillonella. Interestingly, the human papillomavirus infected patient had a the most strikingly disrupted microbiome. However, there was only one sample in that category. So just zeroing in on the Prevotella and the Veillonella, maybe not surprisingly, when we tried to look at the statistical significance of the changes we saw, we didn't see very much significance. The Prevotella increases in average Prevotella seemed to be driven by outgrowth in individual samples, as well as the Veillonella. We did note that there was a significant increase in Veillonella in the HIV-infected young group. In addition to the bacteria that are predominant in the oral microbiome, we saw lots of changes in genre that are on the lower end of the scale, and I'm including Orobacterium and Moriella in this group because they are kind of connected. Every time we saw Moriella increase, we saw Orobacterium increased, and these are pathogens, but not much is known about them, except there's been a couple studies that show that these are increasing in patients with tuberculosis. So the viruses that we looked at so far have been Epstein-Barr virus. We didn't see group changes specifically, but we did see significant changes or trends towards significant changes in the HIV-infected groups, both the young and aging. We did not see these types of differences, however, with the fungus Candida albicans. So just to kind of wrap up the talk and summarize where we're at this point, I've combined all the data that we've accumulated so far in this sort of, it's a correlation heat map. And there's kind of a lot of information all crammed into this one little heat map, but since I'm getting to the end of the talk, I'm just going to point out that if you look in the upper left-hand corner, both the Prevotella and Veillonella increases that I've been talking about were correlated statistically significantly with viral loads. And so although our groups are small, we have definitely made some interesting observations that lead us to look at some more samples. And we've seen some age-related and HIV-associated dysbiosis in the salivary microbiome, and they appear to share some similar characteristics. And interestingly, these, although I didn't have time to point it out real specifically, appear to be maybe even enhanced in patients on ART. The different types of oral opportunistic infections that we looked at all displayed different patterns of dysbiosis. But the most important takeaway message, I think, is to get a clear understanding of the significant changes in the salivary microbiome that are associated with aging or HIV infection is going to require analysis of a lot more samples. But fortunately for us, the WISE has an enormous, enormous supply of biobank saliva samples. So that's where we're at right now. We're in the process of getting more samples and moving this study forward. So with that, I'll stop and I'll take any questions. Yes.
All right. Did you look at any of the functionalities of the bacteria? Because the Veillonella, that's a lactic acid fermenting, isn't it? I mean, did you see physiological or functional readouts of the altered microbiome in the mouth?
Excellent question. I sort of secondhand have seen it, so I did not do any direct experiment.